Just a reminder that there is going to be a review session tonight. AJ and Eli have very kindly agreed to conduct one for you. That'll be in this room, so you don't even have to find a new room, 6 to 7. So you're certainly warmly invited to that. A reminder of the exam setup that you need to bring. You need to bring your ID with you. You need something with your university number on it, if at all possible. And that the rooms that will be split between are different rooms now. It's an odd one or Beisner. And that the name split is just a little tweaked from the last time. Be ready to show the proctors your ID so they know you're at the right room. We don't have a lot of extra space in either of the rooms for people who've gotten to the wrong room. Are there any kind of procedural questions about either the review session tonight or the exam on Wednesday? Sorry? You're going to make the PowerPoint available? It'll probably be posted at 6. Okay? Is that right? Yeah. Okay. Any other questions about? Yes?
Will there be a video of the review session?
I don't believe so. No. No video. Other question?
Will we need calculators for the exam?
No, you do not need calculators for the exam. Other questions? I'm not going to ask you to interpolate numbers on a line, so you can call about that. Any other questions? Okay. And going back to what we were talking about, we started talking about the citric acid cycle, and a student made a correction for me. He did so after class, and I would like to encourage you that if you see me saying or doing something wrong, to speak up at the moment so we can correct it right away. So I'm going to make I'll say this correctly this time. It was correct in the handout, but I said the wrong thing, okay? Certainly the citric acid cycle is an amphibolic pathway, that it's both catabolic, breaking things down, and anabolic, providing precursors to build things up. And some of the intermediates, the compounds inside the cycle, are precursors for other compounds. And we mentioned that succinyl-CoA goes to heme, and all organisms, as far as I know, need heme. Almost all of them make their own. Alpha-ketoglutarate can go to glutamate, and that's what this reaction is here. Here's alpha-keto, so glutaric acid has 2 carboxylic acids. Alpha-ketoglutarate has the keto group, the carbonyl oxygen, at the alpha position. And it can be reduced and have an ammonia added to make glutamate. Now, glutamate, of course, has the abbreviation E, glu-E, right? So the abbreviation for glutamate is E, and the product of this reaction is glutamate, or E. It consumes NADH. So this is one of the few cases where you see something that looks synthetic using NADH rather than NADPH. And then the glutamate can go on and be used for glutamine. Q is for cutamine, glutamine. And we'll see those reactions when we get to nitrogen metabolism. And it's the glutamine that is the precursor for the bases in the nucleotides. So I hope that clarifies my slightly confused presentation on Friday. Any questions about this before we move on? Okay. So we're sort of entering into metabolism through the carbohydrates and the citric acid cycle. What we're going to see is that the citric acid cycle intermediates can go out into all the pathways. So in addition to being able to burn anything that goes into it, we're going to see how amino acids and fatty acids get into it, but it can burn all the major fuels. It goes outward, providing intermediates to build up other things. So citrate, we're going to see in a little bit, is a precursor, can be used as a precursor for cholesterol. We make a lot of our own cholesterol. It's not just our diet. It's a precursor for fatty acids like the palmitate and so forth that are the precursors for phospholipids. We've seen already how oxaloacetate can be withdrawn to make glucose. We've seen how we can also make a pyruvate molecule out of a malate, and pyruvate can in turn be used for a variety of things. You wouldn't, you wouldn't backtrack to oxaloacetate at this point. You just make it out of the malate. But you can take the pyruvate and make other things out of it. You can use the succinyl-CoA, as I said, for heme, and the alpha-ketoglutarate through the glutamate and the glutamine to a variety of amino acids and nucleotides. So a very, very flexible, central pathway for a great deal of metabolism. Now, it helps to keep in mind that this relationship between the citric acid cycle and its anabolic role, and to some extent its catabolic role, depends on there being some way for communication between compartments in the cell. Now, in bacteria, they have a citric acid cycle and they only have one compartment, so they just kind of muddle along. We're eukaryotes, and our cells are more complicated. And so in order to get the right things to be happening at the right place, eukaryotic cells have shuttles that enable those compounds to move around. So let's take a little bit of a look at that and what kinds of things are going on. So we mentioned, for example, that citrate might be used for lipid synthesis. We can withdraw it from the matrix of the mitochondrion out into the cytoplasm, and using a separate enzyme, ATP citrate lyase, generate an oxaloacetate and an acetyl-CoA. And the oxaloacetate has still carbons in it that might be useful. A separate malate dehydrogenase, so this is a malate dehydrogenase in the cytoplasm, makes a malate. And a special enzyme called malic enzyme can turn malate into pyruvate. So here we have a malic enzyme. Malate is the 4-carbon backbone dicarboxylic acid with an alcohol. And you can reduce the NADP+, generating a pyruvate and a carbon dioxide. And those 3 carbons can go back into the mitochondria now and re-enter the citric acid cycle sort of from the back side. So this is a way of getting citrate molecules out into a form that's useful for lipid synthesis. We're seeing different pathways in different parts of the cell. So in the cytoplasm, we've already talked about glycolysis and gluconeogenesis. They're both in the cytoplasm. We're going to see that in some cases the anabolic and catabolic pairs are in different places. So this is showing you that lipid biosynthesis is out in the cytoplasm. The lipid breakdown is happening in the mitochondrion. And there are a number of other examples of that as well. But this shuttle would be used for this purpose at a time when you had an abundance of energy in backbones, and so you wanted to store some of them in the form of lipids, for example. So the fact that it's costing you some energy here and costing you some energy there, that's okay because you're in an energy abundant status, and so spending it on making lipids is a way of saving for the future. Any questions about this before we go on? Okay. So this presents us with another aspect of integrating the metabolic pathways. It's easy enough to think of the citric acid cycle as a catabolic pathway of burning fuels that are fed into it and coming out as carbon dioxides. But if we take intermediates out of the citric acid cycle and use those to make lipids or amino acids or whatever, we could use up the whole citric acid cycle at some point. And so if we're going to use the citric acid cycle in an anabolic way, we have to have some way of adding back into the citric acid cycle new carbon backbones. I mean, one way of sort of organizing this in your head is to think about the carbon backbones and what they're doing. And so if we pulled some out to build fat or to build amino acids with, we have to throw some other carbon backbones in at some point. And these reactions that can restore carbon backbones to the citric acid cycle are called anaplerotic reactions, filling up reactions. So these are reactions by which things can get put back into it. And perhaps not too surprisingly, the places at which you feed things back into the cycle bear some relationship to the places where you take things out. So, for example, the reactions that allow you to take carbon backbones from amino acids and put it into alpha-ketoglutarate. Other reactions allow you to feed carbon backbones from amino acids in at fumarate. We've seen how you can feed pyruvate into oxaloacetate. There are other amino acids as well that can add carbon backbones there. Some amino acids come in at succinyl-CoA, the 4-carbon backbone, and we're going to see that odd-chain fatty acids get fed in at that point even chain fatty acids can get burnt up completely. So they don't provide any useful carbon backbones. We know that the pyruvate can get in here by the reaction that we've seen before, pyruvate carboxylase. Remember, we saw that reaction when we were talking about gluconeogenesis. It can also be used to refill the citric acid cycle because the oxyl acetate looks the same. Do notice that basically what we're seeing in here are mostly amino acids being fed in. The pyruvate can enter in. A little tiny bit of fat can get fed in. But each of these are reactions that can raise the overall level. If you raise the level of alpha-ketoglutarate, because it's a cycle, in a very short time, the levels of all the intermediates will go up. So it doesn't matter a whole lot at which point you add it in. You can regulate the activity of that pathway at any of those points by changing how much is coming in. So these are some of the reactions that get used as anaplerotic reactions. These are the reactions that refill the citric acid cycle. And these reactions are especially valuable if you're going to be pulling intermediates out for synthesizing sugar, for example. So for instance, if you have an aspartate molecule, it's a four-carbon backbone. It's actually a dicarboxylic acid already. And a reaction called transamination allows us to transfer this nitrogen to the carbonyl oxygen. They just kind of switch places. And now you have a four-carbon backbone with a carbonyl oxygen, which we recognize as oxyl acetate, and a three-carbon backbone with the amine group, which is alanine. So you can think of this reaction in several ways. You can think of it as a way that aspartate can be used to make more oxyl acetate for the pathway, or you can think of it as a way to make alanine out of pyruvate. Kind of depends on the needs of the cell at the moment. But now we have a molecule of alanine we didn't have before. There's another sort of broad category of reactions that helps make it possible to get carbon backbones from amino acids into the cycle. It uses an enzyme called alanine aminotransferase. And this, again, has some resemblances. You've got your carbonyl oxygen here in pyruvate. You've got your amine group in the glutamate. And the alanine aminotransferase just transfers that nitrogen over. And so now you have the amine group making an alanine, like that. And now you've got an alpha-ketoglutarate. So this, I think, should start to bring home to you how close the dicarboxylic acids are and to the various amino acids, that they can be related through that carbonyl oxygen and the amine groups very easily. Now, if you can pull all of these backbones out of the citric acid cycle, you might think that we could break down fat and feed it into the citric acid cycle and take it out as sugar. We can't do that. We can't do that because in our bodies, for the citric acid cycle, every time you feed in an acetyl-CoA, you burn off 2 carbon dioxides. So feeding lipids into the citric acid cycle provides energy, but it provides no carbon in a usable form. It's all coming out as carbon dioxide. Plants, on the other hand, are very good at making sugar from fat. And that's a good thing because sprouting seeds often keep their energy in the form of fat and want to make it into sugar to get on with their daily business. So plants have a relative of the citric acid cycle called the glyoxylate cycle in a different organelle called a glyoxysome. And the part of it looks very similar. You've got oxyl acetate, your 4-carbon dicarboxylic acid, and acetyl-CoA, which can come from fat. We haven't seen those reactions yet, but please trust me, you can get acetyl-CoA out of fat. And those two together give you a citrate, which makes isocitrate the way it does in us. But what the plants do differently at this point is they have an enzyme called isocitrate lyase. And isocitrate lyase it just goes straight to a 2-carbon fragment, glyoxylate, and succinate. So the rest of the glyoxylate cycle is just like the citric acid cycle. You've got succinate, fumarate, malate, oxyl acetate, some NADH, and that kind of thing. But now you have a 2-carbon fragment that you can do something with. And so instead of losing 2 carbons as carbon dioxide the way you would in citric acid, instead you capture it as a little 2-piece glyoxylate. Now the glyoxylate and a second molecule of acetyl-CoA through an enzyme called malate synthase, because it makes malate, produces oxaloacetate. So 4 carbons of this isocitrate end up in this oxaloacetate, and 2 carbons of this isocitrate end up in this oxaloacetate, with the acetyl-CoA providing the other 2 carbon backbones. So we're feeding in 2 carbons up here, 2 carbons up here, and we get out of the whole process an oxaloacetate molecule, where before we only had a bunch of acetyl-CoAs. So this oxaloacetate then, for example, could be used for gluconeogenesis. This trick won't work in us because we can feed acetyl-CoA into the cycle, but we're going to burn off both the carbons as carbon dioxide. Is this clear? Are any questions about that? Okay. Now, one of the valuable things about the citric acid cycle is it was in the context of the citric acid cycle that biochemists came to understand how enzymes treat molecules asymmetrically. You remember we talked back at the beginning about how if you have 4 different residues on a carbon atom, that they can come in 2 forms, left and right-handed. And yet, enzymes always seem to work with the same consistent handedness all the time, whereas if you do many reactions in the test tube with inorganic catalysts, you get a mixture. Well, the citric acid cycle helped give us insight into how that works. Now, this was the original way that Krebs proposed for it to work, that oxaloacetate and pyruvate would make a citrate, and there would be an aconitase intermediate that somehow the cells could just simply use pyruvate itself, so there was no acetyl-CoA, no CoA involved. And if this cycle had been true, it suggested that you would get an absolutely symmetrical distribution of carbons in your isocitrate, because this is a 2-carbon fragment, that's the 4-carbon fragment, and in the citrate, it looks symmetrical, certainly. There's nothing about the structure of a citrate molecule that looks like you could tell the 2 ends apart. And so they assumed that you would be getting it out symmetrically afterwards. But that wasn't what they found. That wasn't what they found. And this shows you in a little more detail why they initially thought that the carbon dioxides would come off symmetrically. So if they fed in oxaloacetate, where you could tell where the carbon was. So you put your radioactive carbon on the end that's farthest away from the carbonyl oxygen, or carbonyl carbon, and you use that to make a citrate molecule. Now you've got a citrate molecule that has a radioactive carbon, and you know that that radioactive carbon came specifically from oxaloacetate. Now, if the enzymes had no way of telling the overall structure of things, what you would expect is that when the enzyme used that citrate to make a conatate, that half the time it would end up, farther away from the double bond, and half the time near the double bond. And then if that happened, you would then expect one specific end, one end to get it in some of the molecules for isocitrate, and the other half would get it at the other end. So the prediction, in the absence of knowing how enzymes worked, was that if it went in through oxaloacetate, it could come out either way. But that was not what was happened. In fact, the only thing that they saw was a labeling of the alpha-ketoglutarate at one specific end of the molecule, not as a random mixture. That was very, very puzzling. It was very puzzling, because citrate is clearly a symmetrical molecule. So the enzyme can see something about citrate that we can't see. Let's see. So they did some hand waving for a little while, and they wanted to suggest that maybe the first thing that was made was a conatate, and that there was some side reaction that scrambled into acetate, and then that came down to isocitrate. But that actually didn't solve their problems. What really was going on is the fact that when the substrate binds to the enzyme surface, in the process of binding, it is restricting the freedom of the different groups to do things. And so it makes the groups non-equivalent. And this is a schematic way of trying to represent that that if this molecule is the citrate molecule, you've got the symmetrical back ends, if you like, and you've got a hydrogen and a carboxylic acid here. And when these two groups bind and sit down on the enzyme, this other carboxylate group would be indistinguishable from this one out in solution. But now that it's sitting on a surface, this one is the surface-bound one, and this one is the one that's sticking out away. So one way that the physicists would call this is they say that the binding to the enzyme broke the parity, that the symmetry of the soluble molecule is lost when it binds to a substrate because the substrate constrains what can happen. So the carbon that comes in at citrate synthase is in one particular end of the molecule up until succinate. And then succinate is again a symmetrical molecule, as citrate was, but it's one that does not have 4 groups coming off those carbons. If you go back and think about the structure of citrate, it has 3 groups coming off the carbons. And that means that this kind of an analogy breaks down, and it does become randomized at that point. So there are some microorganisms that have a slightly different way of doing things. In fact, if you're interested in these pathways, the amount of variety among the prokaryotes is sometimes astonishing. I mean, you know, we talk about glycolysis, and there's like half a dozen different ways of doing something like glycolysis in prokaryotes. And we see that kind of variety here at the citric acid cycle as well. So in an organism that's missing alpha-ketoglutarate dehydrogenase, for example, using a slightly different enzyme, they can go from alpha-ketoglutarate, so it's alpha-ketoglutarate, to succinate semialdehyde instead of succinyl-CoA. So this is a free molecule now. And that reaction is driven by loss of a carbon dioxide. And then the succinate semialdehyde is reduced to succinate, generating an NAD at that spot. And you might find it interesting to ask yourself how this incomplete citric acid cycle compares for energy yield to the normal complete one that you and I have. So I think, let's see. Right. So we have a little bit more on this. We had some slides on the glycolysis and carbohydrate handout that we did not have time to get to then, and we can go back to those now if you'd like to do that too. But let's take a moment and talk about this particular slide. Talk between yourselves and see if you can arrive at an answer about which kind of an inhibitor this is. All right? Do folks feel like you have a bit of a consensus yet on what kind we have, or you still need some time to settle it in your heads? Does anyone want to tell me what kind of a graph this is called? What kind of a representation of enzyme activity this is sometimes called? One of the names for it is a Lineweaver-Burk plot, after the people who came up with it. And there's another name for it as well that's more descriptive of it in its own right. Anybody remember that one? I heard half of it up there. Thank you. Yes, it's a double reciprocal plot is another name for it. Why is it called a double reciprocal plot? Do you want to spell that out for us? Thank you. Right. So in other words, of the initial velocity. This is not this axis is initial velocity. It's not Vmax. So in other words, this is 1 over the velocity of the enzyme as a function of 1 over the substrate. Right. And so out here is actually quite a low concentration of substrate, and this would be a higher concentration of substrate because it's a reciprocal plot. So what kind of an inhibitor would we say this is? Can we read any of the kinetic parameters off of this graph? Sorry? All right, so you're proposing it's a competitive inhibitor. And okay, so you're proposing it's a competitive inhibitor because this is the number is the same, and so you're saying this would be then the Vmax, is that right? Okay, so because being 1 over velocity, that's the largest velocity, and they're the same there. And then the extension of the apparent Km out there. So again, this line is intersecting on the 1 over substrate thing closer to 0. So that's a smaller, this would be a larger number, and then this would be the smaller number, so that in the absence of inhibitor, you have the greater affinity, the smaller Km. Okay. Any other questions about doing this kind of a double reciprocal analysis? Comments? Questions? Okay. We didn't get a chance to finish the handout on the carbohydrates completely, but I think there's some material in there that's of interest to people in general. Oops, that's anatomy. I don't want anatomy. There we go. So if you have your Chapter 10 handouts with you, I hope. Okay, let's open it. And great. Okay. Are there any other questions about carbohydrate metabolism that we did cover before I go to the supplementary material? Are there any questions you have about glycolysis or gluconeogenesis or pentose phosphopathia, any of those you'd like me to go over or explain some more? Okay. So if not, I mean, I'm happy to go back to other slides if people have questions, but if not, let's think a little bit about the things here. What I'm adding in these slides is some material that goes a bit beyond what's in the textbook, because these are questions that a lot of people engage with in daily life, and so I thought I would try to see how they relate to the material that's in the textbook. These are all examples of molecules called sugar alcohols. Now, you know that sugars are polyalcohols already, so why would these things not just simply be called sugars rather than sugar alcohols? What's different between these things and something that's a true, authentic, actual sugar? Exactly. Oh, give them out a balloon or something. Thank you. There's no aldehyde and no ketones. So there's no carbonyl oxygen. There's no carbonyl carbon. And if you do the math on it of the carbohydrates are called CH2ON. And to make those numbers come out, one of those carbons has to have a double bond to an oxygen. It's either an aldehyde or a ketone. These sugar alcohols are all alcohols. There's no carbonyl carbon anywhere in them. So it changes their chemistry a bit. Not a huge amount, but a bit. but it definitely changes how they're metabolized. And they've become of more interest recently because of people's interest in whether or not dietary carbohydrates have a role to play in overall health maintenance, weight maintenance, and so forth. I'm sure you've heard about low carbohydrate diets. Atkins diet is one of them. And the notion behind low carbohydrate diets was that you would not release so much insulin. Insulin, we're going to see, is a regulator of fat metabolism as well as of carbohydrate metabolism, and that this would help you keep your body from getting so excited about the fat and so forth. Initially, there was some concern about the low carbohydrate diets, that ketosis was somehow rather bad for you. The really kind of energized low carbohydrate type people actually monitor to see if they are in ketosis, and we'll see when we get to lipid metabolism how that differs in energy status from carbohydrates. It does definitely lead to lower insulin levels. That's sort of the point of it. It's probably not as bad for people as initially the mainstream nutritionists thought. One of the things that tells us that low carbohydrate diets are probably not that bad for you is that in the early part of the 20th century, it was discovered you could control epilepsy in many children with a low carbohydrate diet if it was severe enough to keep them in ketosis. These children develop normally. They gain weight, they develop, they become sexually mature, their brains are okay, and they stop having epileptic seizures. Even today, if someone has resistant enough epilepsy that they can't control it with the normal medications, a ketosis diet can, for many of those people, control their epilepsy. So probably not as bad as it was thought, but it is somewhat restrictive, trying to maintain total low carbohydrate. That's given rise to a refinement of this, looking at something called glycemic index. And the glycemic index is a matter of the amount of carbohydrate that's in the blood over time. And we're going to be looking at that, I think, in a little more detail when we get to integration metabolism. And so if something releases sugar into the blood more slowly over a longer period of time, it will have less of an effect on the peak height and the area under the curve that the insulin and the glucose is going to do. Roughly speaking, the kinds of things that tend to have lower glycemic index would be things like whole grains instead of pure ones. Fruits, except for bananas. It turns out that bananas are basically candy, except for their potassium, which is good stuff. Vegetables are pretty much safe going. Most vegetables are pretty low glycemic index. All of these things are relative to the actual demands you're putting on your body. So, for example, someone who's a runner who's running 5 to 10 miles a day, they don't need to worry about any of this. If you've ever watched the Tour de France, you see the bicycle riders picking up double handfuls of French pastry, and they're barely keeping up with their energy demands. So it's always relative to your actual demand on your body. This starts to sound kind of complex, and how do you think about it? And a nutritionist who was working with an art student came up with an extremely simple rule of thumb, which is eat colorful food. If the food's not colorful, it probably has a high glycemic index. So, oh, darn. Now, does this come out on your handout? Because it's... This showed on my screen, so I can't give you the... If you want to see the glycemic index for these foods, you can search on the glycemic index. And I'm sorry it doesn't show up here. But the notion here is that you're comparing foods to glucose. And if you take in 50 grams worth of sugar, of glucose in the food, and then you see how high or how low your blood sugar goes as you're eating those things. And you can see here, if you're using absolutely pure glucose as your standard of 100, here's orange juice at like barely half that, and here's an apple that's much lower still. Even though these are being compared all on 50 grams of glucose in the food. You can see that different cereals can be different, so cornflakes are more like candy, if you like, than Cheerios are. Raisins are almost like candy. But this is on glycemic index now, how rapidly 50 grams of sugar in that food makes your blood sugar rise. Now that does leave out the fact that you eat various amounts of these things and they don't necessarily have 50 grams of sugar in a serving. And so the idea of glycemic load, which was how the sugar tends to elevate the blood sugar on a standardized basis, was modified to the notion of glycemic load. And glycemic load takes into account the glycemic index, the inherent tendency to raise blood sugar, and how much sugar is actually in a typical serving. And so the combination of those two things gives you a glycemic load. And it can make a very big difference. Watermelon is a good behavior again. It's down around a glycemic load of about 10. So this is glycemic index over here, and then this is glycemic load over here. And so if you can see that this has been, you have here, here's spaghetti, carrots, and oranges. Their glycemic index is almost identical. That is, if you eat an amount of those foods that has 50 grams of glucose in it, how will your blood glucose respond? And it'll respond pretty similarly, not as fast as if it was pure glucose, but, you know, moderate response. But if you eat a cup of spaghetti, that's got 140 grams of glucose in it. If you eat a large carrot, that has 72 grams. And if you eat a medium orange, it has that. And so the glycemic loads are quite different, aren't they? So the glycemic load of the carrot is 1/5 the glycemic load of the spaghetti. As a rule of thumb, the nutritionists suggest that people think of below 10 as a low glycemic load, 10 to 20 as a moderate load, and 20 and above as a high glycemic load. And so you can see things like bean sprouts are essentially free. You know, pizza, compare, for instance, here we go. Here we have things that are all in the 50s, right? All of these foods are in the 50s. But the banana ends up with a load of about 14, whereas the potato chips end up with a load of about 30. And that's because the typical serving size of a food has different amounts of glucose in them. These weights here are the weights of the food, not of the glucose, because a lot of the banana is water. Okay? They're giving you the weight of a typical serving. So glycemic load takes into account how you actually eat a food. And if you want to get into this in a more serious, focused way, glycemic load is probably a more useful number to think of. Anything that brings watermelon back into the list of good foods has to be welcomed. There are some other things that are used by people who are trying to avoid carbohydrate metabolism. And this is one example of that. This is what Splenda looks like. Splenda is the brand name for something that's generically called sucralose. And sucralose was developed because aspartame, which was an artificial sweetener before it, generated some concerns in some people. And besides, if you have a new one, you can sell it for a while. And really the only difference between Splenda and sucrose, because it should look a lot like sucrose to you, is this chloride atom up here. So that chloride atom does not interfere with binding to the sweet receptor. In fact, it's somewhat sweeter than the average sucrose molecule. That's why you don't need to put as much in. So it's sweeter than a typical sucrose molecule, but it can't be metabolized. And so you might expect that it has a smaller Km for binding to the sweetness receptor because it takes less of it for the same subjective experience, but it has 0 Vmax for being metabolized into a usable sugar. Any questions about that before we move on? Okay. This is an example that helps you sort of tie together some of the things we were saying before when you're talking about protein structures and a way to concretely think about how these pathways interconnect and how the regulation of these things works. So here is, in the red, the oxygen binding curve of a molecule of hemoglobin. So this is the oxygen concentration here and the oxygen binding curve for that. What you're seeing in line A and line B are individuals who genetically are deficient in one or another enzymes. And these are enzymes of glycolysis. You can see hexokinase or pyruvate kinase are the two enzymes that we're going to be talking about here. And the reason it matters is this little side trip over here to make 2,3-bisphosphoglycerate. If you remember, 2,3-bisphosphoglycerate was the molecule that bound into the middle of hemoglobin to modify oxygen affinity. And so if you stop to think about the connection of the rest of glycolysis to this, you can see that if you were getting rid of hexokinase, or if you're getting rid of pyruvate kinase, you're going to affect the concentrations of 2,3-bisphosphoglycerate. So before we nail it back onto the hemoglobin molecule, let's start with a slightly easier question, which is, which of those enzymes do you think would raise 2,3-BPG levels, and which of those two enzymes do you think would lower 2,3-BPG levels? So why don't you talk about that for a second and see if you can come to a consensus about that. Which of them would raise the levels of 2,3-BPG? Which of them would lower the levels of 2,3-BPG? Anyone feel like you want to take a stab at the effects yet? Anybody want to take a stab at whether hexokinase would raise or lower the concentration of 2,3-BPG? Lower? Okay. So we're talking about deficiencies again. And what would the effect of the pyruvate kinase be? We do have two lines on the graph. Let's explore it for the moment. We'll go on from there. So I didn't quite see who it was that suggested that hexokinase would lower the concentration of 2,3-BPG. Who was that? I won't put you on the spot, but sorry about that. Would anybody like to unpack why hexokinase might lower the concentrations of 2,3-BPG? Yes. Thanks very much. And just so that folks can hear you, I'll repeat it for the people who are watching rather than here. That if you don't have the hexokinase, it's going to reduce the glucose 6-phosphate, and that decrease in concentration just propagates down to the 1,3-bisphosphoglycerate. There are some ways to get these compounds off of side chains, like pentose phosphate pathway, for instance, but certainly hexokinase is the principal source of it. So it's going to decrease it. You might not wipe it out, but it will decrease it. So why would getting rid of pyruvate kinase increase the concentrations of 2,3-bisphosphoglycerate? Right. Pyruvate kinase is the end step, so if you can't complete that end step, the stuff downstream of this is going to build up. It'll back up through the pathway, and you'll have more 2,3-bisphosphoglycerate. Right. That is, anybody feel like they remember their hemoglobin well enough to make a suggestion about which of these two lines is which enzyme deficiency? Okay, so now you're proposing that this would be A. Okay. And would you like to explain why lowering the 2,3-BPG would give you the increased oxygen efficiency or binding? That's right. Right, right. So 2,3-BPG is an allosteric effector. It binds there at the center of hemoglobin, and it tends to favor the deoxy form. So if you can't make as much of it because you're hexokinase deficient, that will, on average, increase your affinity for oxygen. And then by complementary kinds of reasoning, you can find pyruvate kinase giving this phenotype. So here's an example of where changes in glycolysis can change things that we don't think of as being part of glycolysis that could have very major phenotypes. I mean, this person here, individual B, is going to have a very different capacity for carrying oxygen to their tissues. Well, it's pretty close to the end time. I'll see you all Wednesday. Good luck. AD? Okay, I'll let you have it.